Welcome to the video on plant DNA barcoding. DNA barcoding is a specific technique. Identification of species is done based on a short segment of DNA, short segment of a gene which is there in a specific region of the organism. For example, plants are identified based on two small genes that are there in chloroplast genome. Only based on small sections of DNA of about 400 to 800 bases that is present in the chloroplast genome, a plant species can be identified. And this gene sequence that is there in the chloroplast genome is barcoded, and this barcode is used in the future to identify a species. So in this video, I will explain about what is DNA barcoding. Recently, I was a resource person in a RUSA sponsored webinar conducted in Patkar Warde College of Mumbai University. And in that webinar, I gave a talk on plant DNA barcoding. Sponsored webinar called BioFACET, organized by Patkar Warde College of University of Mumbai. First of all, I must thank all the organizers. Especially, I must thank Dr. Ruchita Delvi, who has been in touch with me for a long period of time. Thank you very much for each and every one. And let us start with this topic called plant DNA barcoding.
It gives me immense pleasure to welcome our guest speakers of the day, Dr. Giridhar Parvatam and Dr. Jayarama Reddy.
Good morning, all. It's my great honor and pleasure to introduce the speaker for the second session of today's BioFACET, Dr. Jayarama Reddy, sir. He is a professor and the head at St. Joseph's College, Bengaluru. He had conducted research at IIHR and IISc Bengaluru for his PhD. Sir received his PhD in the field of biotechnology of orchid in 2002 from University of the Bangalore. He guided 4 PhD students so far and currently guiding another 2. He had collaborated with the Wageningen University, Netherlands, for the research in gene cloning and bioinformatics. So far, he had published more than 92 research papers in renowned scientific journal and authored 9 books. He had revealed and deposited structure of the cancer-related protein MAGE-E1 in Protein Data Bank. In 2015, National Environmental Science Academy honored him with the Eminent Scientist Award 2015. St. Joseph College, Bangalore presented him a Scroll of Honor Award for his scientific achievement in 2016. He is a recipient of G.R. Patel Dronacharya Award for Academic Excellence and J. Agarwal Gold Medalist Award from the International Society of the Gene and Cell Therapy. He also awarded by the Puraskar Award in 2021. It's our pleasure to have you here, sir, for enlightening all of us by your knowledge. Thank you very much, sir, and over to you, sir.
Welcome to this RUSA sponsored webinar called BioFACET, organized by Patkar Warde College of University of Mumbai. First of all, I must thank all the organizers from Patkar Warde College who have invited me to be the resource person in this RUSA sponsored webinar. Let us start with this topic called plant DNA barcoding. DNA barcoding, the identification of the plant species or animal species or any organism, is done by using a short section of DNA from a specific gene or genes. The premise of DNA barcoding is that by comparison with the reference library of DNA sections, an individual sequence can be used to uniquely identify an organism to the species level. We create a DNA barcode for each and every plant. So you can barcode a plant. In the sense, the plant is converted into a simple barcode, and the barcode is available in a barcode library. There are so many libraries. One is bold, and you deposit the barcode that is in a barcode library. Next time when you want to identify the plant, what you do is isolate the DNA from the plant. It may not be from the plant, it may be from the environmental sample, it may be from the fruit, it may be from the leaf, it may be from any part of the plant. You isolate the DNA, all right, and then do barcoding and compare that with the reference one. All right, so you are not using the entire plant. You don't require a flower to identify the plant. You don't require fruits to identify the plant. We require only a small amount of material, plant material. Minimum requirement is 2 grams of material is enough to identify a plant, and from that you isolate the DNA, and that DNA you convert into a barcode, and that barcode you compare with the reference barcode which is available in the database, and by the help of which you can identify the plant exactly without much difficulty, without the problems of understanding taxonomy at all. And that is what is the premise of DNA barcoding. So let us take the plant samples. You can take any plant sample. All right, I told you already you require only about 2 grams of the plant sample. From there, what you do is extract the DNA. How do you extract the DNA? There are so many methods by which you can extract DNA. And then you amplify the DNA and create a barcode. There are barcode creators, online barcode creators are available online. Convert the DNA into a barcode after amplification by using PCR. And thus, what you're doing is the sequence of the plant that is collected from the plant is converted into a barcode. So this barcode is deposited in a barcode library, and that will become the reference. Next time, if anyone wants to identify the same plant, same thing they have to do. They have to isolate the DNA, they have to convert that into a barcode and compare that barcode with the barcode that is there in the reference library, so that the plant can be easily identified. And I have given some examples of some barcodes which are already generated. So I have the example of dendrobium barcode, which is there on the screen. Nividias you have, Spiranthus we have, Telemitra you have. These are the plant species of which you have created the barcode and which are available right now on the DNA barcode libraries. There are so many companies all over the world now, even in India, where the DNA barcode of a particular plant can be outsourced. You can give this job to some biotech company. They do it at the cost of 3,500 rupees per plant. All right, and each and every item that is kept in the supermarket has got a barcode. Earlier we had barcodes, now we have QR codes, and by using the barcodes, the customer can also identify the product. At the same time, even the stock manager or storekeeper also can identify the item. Similarly, plant DNA barcodes are also made in the same way for the known species which are kept in the library, and for the unknown species also we have to create that. We have to deposit in the library, isolate the DNA from the sample. So you require the plant sample or animal sample. All right, amplify the target DNA. All right, and barcode it. And when you are amplifying, you have to use the PCR technology. You have to use polymerase chain reaction, by the help of which you amplify the DNA. All right, and then you again compare the resulting sequence against the reference that is there in the database, by the help of which you are matching the sequence. Now there is one step which is the link between barcode and the reference, that is converting your DNA sequence into a barcode. How do you convert your sequence into barcode? There are so many DNA barcode generators online. These are free tools that are available online, and one commonly used DNA barcode generator is BioRad. You have a barcode. What is being scanned here is the QR code. So QR codes are scanned, which are available now on all the products that we buy in the market. And there is a scanner. The scanner has got a magnet. All right, the scanner reads the barcodes, and all the details that are hidden in the barcode are recorded in the scanner and displayed onto the monitor of your computer. And same thing is going to happen for each and every organism, all right, if it is barcoded. So you are not barcoding the entire genome. We are only barcoding a small section of DNA, which is about 400 to 800 bases, not more than that. And it doesn't show intraspecific variation. That means between the species of the same group, there is no variation in that region, but it shows significant variation in interspecific. Between two different species, it may show variations. So that is what is the important feature of DNA barcoding. So these regions, as I told you already, having less intraspecific variation. Within the species, they should not show any variation, or they should show minimum variation. And they should show a lot of variation in the interspecific. Between two different species, there should be variation. All right, and this is what is known as barcoding gap. But I would like to mention the contribution of Paul Herbert. All right, the terminology, the modern DNA barcoding technology, and the standardization of the method, and identification of species, and potentially allocating unknown sequences to higher taxa. All the initial work, all right, all the primary work of DNA barcoding was done by Paul Herbert. Coming to the applications of DNA barcoding, so the people who can use this is taxonomists. Especially this will become handy for the taxonomists. All right, and it will become easy for identification of the plants. Also, life scientists for identification of study of the organisms. Like you're working on a particular plant, and you don't know the exact name of the plant. You want to know the exact name of the plant, and in that case, exact name can be identified and can be obtained by using DNA barcoding. And if you already know the name and you want to confirm whether it is the same plant or not, at that time also it will become useful. So taxonomists, life scientists use it, and conservationists. The plant that you're seeing on the screen is Dioscorea alata. The current method by which you are identifying a plant, like for example, Dioscorea alata, what you're seeing on the screen, the current method of identification is based on a very complex and very, very complicated method. You can see it on the screen. You have to know the characteristics of leaves, roots, stem, flower, stigma, anther, corolla, calyx, floral diagram, floral formula. Each and everything should be known. All right. Most importantly, the person who is trying to identify the plant should have this taxonomic knowledge, should know the meaning of these terms. All right, there are so many terms, technical terms, of which sometimes I also get confused about the names. What is the meaning of those names? And when you want to identify, you have to enlist all these characters, which is a very complex process. Of course, it is a part of taxonomy, and that is what we are doing in taxonomy. So thus, the traditional Linnaean taxonomy is a very complex process, which is very subjective. And even with all this knowledge, with all the information that you have collected, still you may go wrong. You may identify the plant wrongly. All right. And let us come to plant DNA barcoding method. What we are doing is we are only collecting a small amount of DNA, small amount of gene, and we are barcoding it. And we are identifying the plant based on this DNA segment. All right, and that is what is the biggest advantage of plant DNA barcoding. So the traditional taxonomic method of identification of the plant is complex and it is subjective. DNA barcoding technology, where you can use this technology because it is simple, it is objective. Okay, so that is the comparison between our traditional method of identification of the plant and plant DNA barcoding method, which is the advanced technology, which is going to be the future of identification of the plant. A group of scientists published a paper in BMC Medicine. They worked on 44 herbal products from 12 different companies. So they collected 44 samples produced by 12 different companies, and they found that, all right, out of 44, all right, 30, these are all from reputed companies, all right, were contaminated or substituted. So the consumers are put into risk. All right, the plants that are actually grown in the wild are collected from where the herbal products are produced. But these herbal companies, what they are doing is they are not actually bringing the original plant. And so many places you have contaminants, so many places we have fillers, and there are so many places there are no sequence at all. This is what is the need for DNA barcoding. So you can maybe somehow identify the plant. Like if I say tulsi, you may identify. Aloe vera, you may identify. But if aloe vera or tulsi or Ravulfia serpentina or Catharanthus roseus, if it is coming in the form of a powder, can you identify? All right, but the answer is you can do it by using plant DNA barcoding. And you don't require flowers, you don't require plant, you don't require anything. What you require is even a small amount of powder is enough. About 2 grams of powder is enough. From that you can isolate the DNA, and for that you can identify whether the plant is same or not. All right, now you can identify. So ginseng is a plant which is available only in some parts of China naturally, all right, especially in North China. And it is highly medicinal because it is having anti-aging properties. But the cost of original ginseng is 1,000 US dollars per pound. Very commonly, all right, ginseng is contaminated. Plant products using herbal product using ginseng is contaminated with this fake plant. We have two plants on the screen. On the right hand side we have Panax ginseng, the original ginseng. On the left hand side we have, all right, Panax quinquefolia, which is a fake. And even for taxonomists it becomes difficult for identification, differentiating, because these two plants look similar. Cost in the US market $5,000 per pound. It may be less in India. Now I'm showing you the original saffron and also fake saffron. Okay, there is the difference between the original Ravulfia serpentina, Sarpagandha, highly medicinal plant, and it is actually substituted by, all right, we have Ophiorhiza mungus. Okay. So three types of sources that can be used as samples for plant DNA barcoding or animal DNA barcoding or DNA barcoding as such. And after sampling, the next step is, step number 2 is extraction of DNA. So the third step of the DNA barcoding is PCR. There are several criteria that are involved in the selection of the DNA region that can use as a barcode. I have 3 examples here of the sequences that you're selecting, which are good for you or which are bad for you. The first one is a sequence which is completely conserved. Okay. Second one is no conservation. Third one is partly conserved. And what is ideal is a sequence which shows some conservation, which is good for PCR, good for barcoding. Now, these are the genes that I mentioned. Okay, one is matK. matK is maturation K, maturase K, which is about 1,500 base pair, which are there in the plant chloroplast gene. And this encodes for organelle intron mutase, a protein that splices group 2 introns. That is the job of this gene. And this gene is unique to a particular species. This is not all of a sudden we have to come to this conclusion. Over a lot of time, people have worked and they have come to this conclusion. As of now, this is one of the best genes. The other gene is rbcL gene, which is about 1400 base pairs on the chloroplast genome. After selecting the genes, after selecting the specific region, after isolating the gene, the next step is PCR. This is what is a PCR technology, polymerase chain reaction. I'll not go into these details. So you can amplify the DNA by using PCR technology. Small fragment of DNA material is used, and the sequence length is also about 400 to 800 base pairs. All right. Amplification of eDNA, where the amount of DNA is very less, is even more important. Where the size of the DNA sample is even lesser, it is less than 200 base pairs. The next step after PCR is sequencing. You want to know the sequences. Now, earlier the sequencing was very, very difficult and very, very cumbersome process. The first sequence that was developed was kept in a place where it was about 1200 square foot area. But now the sequencers have become so handy. That means that they are so small. You can see on the right hand side, a man is holding a sequencer in his hand. You can keep it in your pocket. A DNA sequencer is as small as your mobile phone. You can keep it in your pocket. Okay, so this is what is Oxford millipore DNA sequencer. Okay, so this is how sequencing is done now. The DNA fibril will enter through a pore, and as the DNA is entering, passing, there is a scanner. The scanner will read each and every base, and based on the response that is coming out from each sequence, each base, whether it is adenine, thymine, cytosine, guanine, it is reflected on the monitor. It is a very, very fast process. You can do sequencing it very, very fast. So it is very easy, very simple. The last step is called reference library. And these databases contain DNA barcodes assigned to specifically previously identified taxa. Already 5 lakh sequences or plant species have been identified. Sequences are available in the databases. Final step of DNA barcoding is done in a reference library. What is a reference library? Reference library is nothing but DNA barcoding database. The largest DNA barcoding library, International Barcode of Life Project, eyeball. All right. The second largest is called bold. Okay, second largest, which has got 5 lakh bins. These are called bins. Bins are nothing but barcode index numbers. This is bold, barcode of life database. Okay, this is barcode of life database. I've tried to identify the sequence of Oriza sativa, which I did it recently. So Oriza sativa is one plant where a lot of work has been done. You can see here FASTA sequence of the Oriza sativa. This blast. This is the home page of blast. Okay, I've made several videos on bioinformatics. You can go to my YouTube channel. It is in my name only. You can just type in Google Dr. Jayarama Reddy YouTube channel. You can go to my channel. I have more than 210 videos. So you can go to the YouTube and you can watch these videos if you want more information about how blast is done and what is the technology involved in this and how comparison is done. One is identification of species. Next is detection of invasive species. All right, delimiting the cryptic species. You can do diet analysis, food web application, especially barcoding can be done for food safety, bio monitoring, ecological assessment. All right, I also told you what you can do is you can do it in case of medicinal plants, herbal plants, which are widely being used these days. So for exact and precise identification of that, you can use plant DNA barcoding. Right? Thank you very much for listening to me patiently. And I must thank each and every one of you. I must especially thank the delegates for bearing with me. I must thank all the organizers of this RUSA webinar conducted by Patkar Warde College of Mumbai University. Thank you for giving me this opportunity being the resource person of this RUSA webinar.